All right, guys. So this week we are covering the intermediate step between glycolysis and the citric acid cycle. So I'm calling this pyruvate and acetyl CoA. So mainly what we're trying to look at is how we actually make acetyl CoA. Now, that's not the longest process in the world, so I wanted to tack in a couple extra things just to give us a little more length. It will definitely be a shorter lecture this week. So one of the things I wanted to talk about were feedback mechanisms. And so we've talked previously when we talk about enzyme kinetics. The idea of an enzyme acting on a substrate generates some kind of product, right? And then that gets acted on by another enzyme, and you just keep having changes and changes. And the idea that one of the products, so it could be an intermediate substrate or product, it could be an end product, can go back and then inhibit allosterically or activate allosterically one of the original enzymes. And so what we see, for example, in glycolysis, which we just covered last time, phosphofructokinase, or PFK, will add an extra phosphate onto fructose 6-phosphate, forming fructose 1,6-bisphosphate. Okay, so that's one of the first steps of glycolysis, that energy input step. So the energy input phase of glycolysis, where we have to spend 2 ATP to get more ATP and NADH out. If you are actively burning through energy like crazy, you're going to generate a lot of byproducts of that. One of those byproducts is adenosine monophosphate. That means you've pulled both phosphates off of ATP to get the energy out, and you're left with AMP. Okay? So if you have a bunch of AMP around, that means you're probably using up a lot of energy. And so you would definitely want, if I see a bunch of AMP around, that's going to tell phosphofructokinase, hey, you need to do your job because we need ATP. We need pyruvate to make more ATP. Awesome. Now, what else could happen? Well, let's say I don't have a large energy demand, so I have a bunch of ATP sitting around. Okay, it's just waiting to be used up. Citrate, same thing. So citrate is one of the molecules in the citric acid cycle. That's kind of what it's named after. It's that first step. And so if there's a bunch of that sitting around, that means I'm not having the citric acid cycle do its thing. It's not generating NADH to make ATP. And so I don't have a large energy demand. So if I have these products sitting around, then I'm just not using a lot of energy. So it's going to kind of turn off glycolysis. It's going to close that spigot a little bit. Now, it's not perfect. It doesn't completely close the spigot, as if this is like some kind of like faucet or something. But it can slightly close it. And then this one would slightly open it. So that's just the idea of a feedback mechanism. Now, there are other feedback mechanisms throughout glycolysis as well as the citric acid cycle. I just wanted to throw this out there just to kind of expose you guys to just a variety of these steps. And so here on the left side, we have glycolysis. So this is the step I just showed you with phosphofructokinase. Pyruvate kinase near the end of glycolysis also gets controlled by some of the intermediates. So if you have a lot of fructose 1,6-bisphosphate, which we just talked about, that kind of stimulates it, tells it to keep going. If you have a bunch of ATP, well, why in the world would you keep going with glycolysis? Let's stop this whole thing. So ATP not only turns off this step right here, but it also turns up a further along step in glycolysis. Because, I mean, why do I want to keep spending all my time and energy wasted on glycolysis if I'm not going to use the actual byproducts, okay? Now, the other direction also occurs, but it's kind of the opposite. So gluconeogenesis is when we take the byproducts of glycolysis, or just byproducts that are sitting around from whatever, and you're going to take some pyruvate, for instance, which you can get from a variety of places, and then you're going to take that and send it to actually make glucose, which usually that's only going to really happen in your liver, maybe, at least in humans, because we want to form the large starchy polymers like glycogen in your liver. And so in order to do that, you have to have glucose that you then put into the glycogen. And so you control that a variety of ways. So like if there's it's kind of the opposite. So if I have a bunch of adenosine monophosphate, okay, so that means that I have been using a lot of energy up. So if there's a bunch of that around, that means I need to be going this direction. I need to be making ATP. I don't want to be wasting all my energy making more glucose. I want to be breaking the glucose down, not making it. So if I see AMP, that means I'm using up glucose to make ATP a lot, and I should probably keep doing that. So that's just the logic behind all of that. Now, just to reorient ourselves to glycolysis, so we went through all the steps last time. We started with glucose. We used a couple enzymes to add phosphates to our molecule. We broke it in half. We ended up with a couple G3Ps, or GAPs, I'm sorry, glyceraldehyde 3-phosphates. And then we took each of those and sent them through the next few steps of glycolysis to end up with a couple NADHs and a couple ATPs. So we had a net gain of 2 ATP and we had a net gain of 2 NADHs, even though we actually make 4 ATP, but we had to spend 2, right? So that's where we are. And overall, our net delta G is very, very negative, which is awesome. That means that mostly because of all the ATP that we added, we actually make this kind of an overall spontaneous process because of how the enzymes actually function. Now, one of the byproducts that we're going to end up with is pyruvate. And so we're going to kind of track pyruvate. Glycolysis is an anaerobic process. Now, glycolysis will occur whether or not you have oxygen to drive the electron transport chain, okay? So sometimes people will say, is glycolysis an anaerobic or aerobic process? Well, it's anaerobic in that you don't need oxygen to do it, or you don't need your terminal electron acceptor to do it. However, that doesn't mean that if you have oxygen, it doesn't occur. It's kind of always happening. And so if you have pyruvate being generated, well, then we need to do something with it. If I have oxygen or my terminal electron acceptor around, then I will send it through the electron transport chain through a variety of molecules like NADH and FADH2. If I don't have those things, well, then I need to do something with my byproducts. I need to do something with my NADH, and I need to do something with my pyruvate. And so we're going to follow the different fates of pyruvate. Now, there are a few different fates of pyruvate. The one that we're going to kind of focus on next time would be taking pyruvate and sending it through the citric acid cycle, which is where we're going to take acetyl-CoA, which you can see here, and we're going to send that and combine it with citrate, and we're going to send that through the citric acid cycle, otherwise known as the tricarboxylic acid cycle, otherwise known as the Krebs cycle. It's going to be great. We're going to spend a lot of time talking about that. But you'll see there are a bunch of other processes that can take pyruvate and we can do other things with it. So if, for instance, you have to have your terminal electron acceptor, which for humans is oxygen, to actually go through the citric acid cycle. And so if I don't have that, this is not an option, okay, to take acetyl-CoA and send it to the citric acid cycle. So I would have to do something else. So in humans, we go through lactic acid fermentation. So I can make this, okay? If you are a different kind of creature that has the correct enzymes, then you can actually go through ethanol fermentation. And there's actually a few other processes as well along these lines, different fermentations. There are other things that you could do. You could turn your pyruvate into an alanine because they're really not that different, which is an amino acid. You could also send it backwards, like I just mentioned, back this way through gluconeogenesis, okay? So there are a variety of paths, and so we're going to kind of just look at a few of those today. So let's get to it. I really like this. This is from Draw It to Know It, awesome website. I've talked about it before. This is just kind of a more visual way to look at what I just talked about, so it's simplifying it a little bit too. So next time, we are going to focus on going through this process, sending our pyruvate to our acetyl-CoA, which we're going to talk about this step today, and then we're going to go into the citric acid cycle, and that'll be next time, okay? So we're not going to talk about that today, so that's cellular respiration. We say respiration, usually we're thinking about oxygen, but it doesn't have to be, but we'll talk more about that next time. You could also go to gluconeogenesis, which we're not going to talk about this time either, but basically you're just going the other direction, okay? So we're actually going to talk a little bit more about these two possibilities. So you can take your pyruvate and you can send it through lactic acid fermentation, if you're a mammal, basically, or you can send it through ethanol fermentation, which is what yeast, like Saccharomyces, does. There are many bacteria that do that as well, like lactobacilli. And basically, we're going to deal with the NAD plus and the NADH. That's generally our issue. Talking first about lactic acid fermentation. So this is what humans do, okay? So in humans, we take our glycolysis, and what are our byproducts of glycolysis? Well, we get a net gain of 2 ATP, right? And we also get 2 NADHs. Now, in a perfect world where we have the presence of oxygen, because we're dealing with humans, oxygen is our terminal electron acceptor, and so our electron transfer chain is cruising, it can take that NADH, awesome, it can put those hydrogens on the other side of a membrane, and it can make ATP. But we can't always do that. If you don't have your terminal electron acceptor, which is oxygen, then you can't do that. So we need to do something with our NADHs, okay? Got to get rid of that NADH, and that's the main job of these fermentation processes, is what do I do with the NADH? The other job, the secondary job, is what do I do with this byproduct? I need to get rid of pyruvate, okay? I need to get it somehow out of the body. I need to get it away because I need to deal with it. And just building up a bunch of pyruvate is not good. It's going to be toxic, okay? And so what I can do is I'm going to use an enzyme called lactate dehydrogenase, okay? And what it's going to do is it's going to take hydrogens from my NADHs, okay, that I generated from glycolysis. It's going to remove them, making NAD, so that can kind of restore glycolysis, let it keep happening, because I need to make that ATP too. And then what it's going to do is it's going to take those hydrogens and it's going to stick them onto my pyruvate, okay? So check this carbon out right here. So I've got this carbon. I'm going to basically pull one of these double bonds over here, okay? And I'm going to stick a hydrogen onto this oxygen, forming a little alcohol group, and then I'm going to balance out this little carbon here and stick a hydrogen onto the other end here, okay? And that is called lactate. And that is an easier molecule to actually get rid of out of the body. Now, if you are a different kind of creature and you have a couple different enzymes, then you can actually generate ethanol, okay? So this is how you would make like beer, for instance, or root beer, same process. So we still have glycolysis occurring, you're still getting that ATP out, and you're still making that NADH, which you need to get rid of. And so we take our pyruvate, we use our pyruvate dehydrogenase, okay, which is going to come up again in a little while. We're going to talk about basically a different pyruvate dehydrogenase that's going to do a whole nother set of steps. So think about it kind of differently though. And so what do dehydrogenases do? Well, what it's going to do is it's going to remove a CO2, okay? And we're left with this acetaldehyde, or acetaldehyde, however you want to say it. And then we're going to use our alcohol dehydrogenase, right? And we're going to generate an alcohol. And so it's going to dehydrogenate it. So we're basically just going to take some hydrogen. So just like before, when we took our hydrogens and we took them off our NADH and stuck them onto pyruvate, well, we're going to do that again. We're going to take these hydrogens and we're going to stick them onto acetaldehyde. And it's almost identical, okay? When we took our pyruvate here and we took this, made this into an alcohol group and stuck a hydrogen on the end, it looks almost the same, except we're just lacking that carbon dioxide on the end here. So it looks almost the same, right? So we have an alcohol group, hydrogen, alcohol group, hydrogen, okay? We're just missing this guy. So that's the only difference. But this is ethanol, which you can drink and will make you feel really gross and hurt your liver. But so we don't have, as humans, we don't have these same enzymes, so we don't do that process. We have lactate dehydrogenase. Now, I mentioned a little bit about gluconeogenesis. So as I said before, one of the things that can happen is we can go, so we just talked about these two things. We can go the other direction, right? You can follow gluconeogenesis. And so what we can do is go backwards. And so this whole process, so glycolysis is very exergonic. You get variable numbers. I'm so sorry. You get different numbers. So earlier I had like minus 100 kilojoules per mole. This one's minus 85. It just kind of depends on what's going on or what kind of cell you're in. It doesn't really matter. It's a really, really big negative exergonic reaction. So how in the world can you go the other direction? Well, 3 of the enzymes in glycolysis are so exergonic that they're essentially irreversible. And so with gluconeogenesis, if I want to go backwards, I need to use actually 4 different enzymes to go the other direction. Okay, now this is usually only occurring inside the liver if I needed to make glycogen. Okay, so that's why you would do this. So on the right side, I have glycolysis. And on the left side over here are the enzymes that I have to use to actually defy glycolysis and go the other direction. And it's still endergonic, okay? Mostly because I have to spend a little bit of ATP to go this direction, which kind of makes sense. I mean, you got to spend a little money to build something, right? And so we end up with a negative delta G, but that's because we had to spend some ATP. But basically, that's gluconeogenesis. So that's another option for your pyruvate molecule. If you end up with way too much pyruvate and you don't know what to do with it, well, you could turn it into lactate. If you're a yeast, you could turn it into alcohol. Or if you're really, really full, maybe you need to make some glycogen, and so you're going to send it back and make some glucose. Why in the world would you go all the way down to the end to make pyruvate to go the other direction? Well, I don't know. You're doing this like millions of times a second, so I wouldn't get too hung up on that. Now, we're going to go through the next few steps is the big one. This is the main thing that I want to talk about. I want to talk about how we actually get from pyruvate to acetyl-CoA. It looks really simple, right? All we're going to lose is a carbon dioxide and we're left with an acetyl group. Okay, it's not that simple, but I'm going to try and get us there, okay? It's not that bad, I don't think. So pyruvate. So we've got our pyruvate, and we're going to be building this up. So we're actually in the cytosol, and the cytoplasm over here, and that's where all our enzymes are, where glycolysis occurs. So that's a classic question that gets asked. Where does glycolysis occur? Well, it happens in the cytosol, or cytoplasm. I don't really care, either one. Now, what's going to happen is we're going to build up a ton of pyruvate, and so we have a very high concentration of pyruvate on one side, not a high concentration on the other, so that gives us a concentration gradient. And so we can use a voltage-dependent anion channel because it is an anion. So cations are positively charged, anions are negatively charged, and pyruvate itself has a negative charge, and so we can use an anion channel along with another carrier. So it's a mitochondrial pyruvate carrier. So it's we have some non-specificity with the voltage-dependent anion channel and some specificity with the mitochondrial pyruvate carrier. Okay? And those are what's going to allow us to transport through the two membranes of the mitochondria into the mitochondrial space in here, inside that nice area where we're going to be able to do some stuff. On the next slide, I have a better picture just to orient yourself. This one, why am I showing this one first? Well, I just want to kind of show you the basic process of oxidizing a pyruvate, okay? Because we need to pull electrons away, we need to reduce other things, and thus we need to oxidize something else. So we're going to oxidize pyruvate, and we're going to reduce NADH, okay? And that's NAD plus to make NADH. So this is getting reduced, this is getting oxidized. And so overall, what we're going to do is we're going to remove some carbon dioxide, okay, because we're going to have somewhere for the carbons to go. And we're going to take the energy from breaking those bonds, and we're going to make NADH, okay? And we're going to end up with acetyl-CoA, which then basically goes through almost the identical process, just with way more enzymes, and that's the citric acid cycle. Now, how does this actually occur? Well, this big white line here is controlled by what we call the pyruvate dehydrogenase complex, which is a massive complex of enzymes. When I say massive, that's like an understatement. So it's a massive complex of enzymes, E1, E2, and E3. We're going to talk about them and what they do. So let's get to it. The oxidation of pyruvate. There's a lot going on here. I know, I'm talking fast. It's going to be great. Now, here is, so we're kind of zoomed out a little bit, okay? So there's our pyruvate. We're going to get that through with the MPC and the, what is it, VDAC? Yeah, VDAC. So that's what gets us through here. We get our pyruvate. And what we're going to do is we're going to send it to this first complex. So the first complex, they spelled things wrong. This is the best diagram I could find that kind of showed everything. So I apologize. They spelled some stuff wrong, but whatever. It's fine. You can deal. So we've got our E1 enzyme complex. This is all one giant complex, but we've got our E1 portion, which is our pyruvate dehydrogenase portion. And what it's going to do is it's going to decarboxylate. So decarboxylation is the removing of CO2. Okay, so we're slowly going to be taking, we have 3 carbons in our pyruvate, and we need to slowly get rid of those as carbon dioxide, right? If we fully oxidize our glucose, we're going to end up with carbon dioxide and a bunch of energy, basically. And so what we need to do is decarboxylate, and that's what E1's first job is to decarboxylate, and it's going to take what's left over, which is an acetyl group, which acetyl-CoA is what we're going to end up with. So we're going to take that acetyl group, so we're not going to mess with those carbons anymore. We're going to attach it to, we're just going to call it TPP. It has a longer name, but to this molecule called TPP. And TPP is just going to hang on to it and allow us to move it around, okay? And so the other job is to take that acetyl-CoA, which we kind of use the TPP to hold on to it so we can decarboxylate it. And then we use that to then move our acetyl group onto a lipoyl group. Okay, so what is that? A lipoyl is just this really big molecule that has a couple sulfurs on the end. And what we're going to do is we're going to attach our acetyl group. So there's our acetyl group right here. We're going to attach that to one of the sulfurs. Why do we do that? Well, if you recall, we see sulfur a lot. When we've seen sulfur, we call them thiols. So we see those in like cysteine residues. They can form disulfide bonds. Why do we like those? Well, they're covalent bonds, but they're much easier to break because sulfur has those, it's a bigger atom physically. And so it has those outer electrons. And so it's easier to pull things off of sulfurs than it is other covalent bonds. And so what we're going to do is do that. We're going to take our acetyl group and we're going to attach it to one of those sulfurs, okay? To a lipoyl. It's just the name of it, okay? So how this diagram is oriented is the top here is where you start, and then you add the thing kind of at the bottom. So we're going from the top to the bottom, and so you can see where there's our lipoyl with those 2 sulfurs, and then we're going to attach our acetyl group to one of those sulfurs. Now, the job of the E2 complex Uh, it's the, oh man, dihydrolipoamide um, acetyl transferase, which they also spell transferase wrong, but hey, it's fine. So what is that going to do? Well, we need to take that guy, and it's going to take the acetyl group and stick it onto coenzyme A. Now, coenzyme A, I'm going to show you a picture on the next slide. Ignore all the stuff at the top here. Why make extra slides? So this is our coenzyme A, which I've shown you before in previous talks, where there's our adenosine chunk, there's our cysteine end, and then there's the acetyl group. So this is what we're making. So this is the job of the E2 enzyme, is to make this actual guy. And so that's what's happening right here. So there's our coenzyme A, which is actually attached to, it's got the sulfur on the end, and we're going to pull that hydrogen off and stick our acetyl group on there. So we're making acetyl CoA. So it now looks like this. What that's going to do is then go and head into the citric acid cycle. And so we're kind of done with that, which is really, really cool. Now, what's interesting though, is this whole process allows us to reduce, okay, so we've gained electrons, onto those two sulfurs. So look up here. So we've got our sulfurs here. They don't have hydrogens. Then we're going to, through the process of the magic of oxidation and reduction, we're going to reduce those sulfurs to actually have hydrogens attached, which is really, really cool. And why is that cool? Well, what we can do is send it to the E3 complex, which is also dihydrolipoamide, but it's a dehydrogenase. Well, it's going to take hydrogens off, okay? And we're going to take those hydrogens and we're going to stick them onto FAD, which is forming FADH2. Okay, so we have reduced FAD. Okay, so FAD has gained electrons. We are oxidizing those sulfurs. Okay, so we're kind of restoring it back to its original lipoyl ability, or structure. And we're reducing FAD to FADH2, which we're going to see that actually happen in the citric acid cycle as well. So I kind of wanted you to see that. And what we're going to do is then use the E3 complex to then reduce an NAD to an NAD plus to an NADH. Awesome. We've seen NADH before, right? That's what we're going to use, and we're going to send that to the electron transport chain to make 3-ATP. That's pretty cool. And so this whole process, we're doing a bunch of different stuff. But basically, we're just taking our pyruvate, we're oxidizing it, we're pulling that CO2 off, and we're taking the energy from that, basically, to stick it onto a coenzyme A that can then go to the citric acid cycle to be oxidized even more. And we're able to take some of that energy and use it to reduce an NAD to an NADH, which we can then use to make more energy. Super duper cool. Now, the pyruvate dehydrogenase complex is, to say it is massive is an understatement. It has 60 active sites. It's 5 times larger than an entire ribosome. An entire ribosome. Those things are friggin' huge. Okay, so we're gonna take, it's 5 times bigger than that. It's so big. That should blow your mind. It blew my mind. I didn't know about that until I just read it like a couple minutes ago. Simplified diagram of what's actually happening. So we've got our pyruvate making carbon dioxide in the E1. The E2's job is to take the acetyl group and stick it onto coenzyme A. The E3's job is to make NADH. Okay, so there's a nice simplified version of what's actually happening. I probably should have shown you this one first and then the other one, but hey, what you gonna do? Now, here's a picture. You always gotta see a picture. I don't know, every textbook I've ever seen shows you a picture of what the pyruvate dehydrogenase complex looks like. So you can see it looks kind of goofy, right? This is, we're zoomed way in. That's 500 angstroms, okay? So we're super zoomed in. But you can see it kind of looks like that, right? You got that little core right there and these outer pieces. This is a drastic oversimplification. So if you go to the protein database and actually look up pyruvate dehydrogenase complex, they show you this picture and they're like, yeah, this is like, I don't even remember what it was, like 25 out of 100 of the actual complexes that are shown. So it's like a giant dense thing, but pretty cool stuff. Down here, these are just the E2 portions, because that's the paper I got this from is just showing the E2 portions. Anyway, so that is really it for today. So that's taking pyruvate and making acetyl CoA and the energy we actually get out of it. So hopefully that's kind of cool. I hope you like it. So next time we are going to tackle the citric acid cycle. So I'm cutting you a little slack this week because next week it's going to be a lot.